Sir, something. Yes, sir. I have started YouTube. Bas. Right. Pudum.
Have you started the YouTube, sir?
Yes, I have started.
All right. Today, we are very happy to have Dr. Yogesh Soche with us. He will speak to us about microbiome, a very, very exciting field, microbiome. So we're all waiting to hear from Dr. Yogesh. Before that, I would like our president, Dr. Arvind Deshmukh, to please say a few words.
Saudi. Thank you, sir. Thank you. It's really a great honor today. One of my friend and one of the talent microbiologist of India, Dr. Yogesh Soche, who has a very great contribution in gut microbes and other fields also. And he is on our platform. So instead of thanking Dr. Yogesh, I will first thank to Dr. Swarnajit Singh, sir, because he is a key person to make this event. And then I will go to Dr. Yogesh, sir. Thank you, sir, for coming on this platform. I'm sure that whenever your lecture is recorded and will be circulated, many more will take the benefit from your lecture.
Yes.
Yes. Thank you, sir. Thank you again.
Thank you, Dr. Deshmukh, for the nice introduction. And now I call Bhupinder Sharma to welcome Dr. Yogesh.
Thank you, sir. First of all, am I audible, sir?
Yes.
Yes, yeah.
Okay, sir. Good morning, one and all. On behalf of MBSI Chandigarh Tri-City, I, Bhupinder Sharma, welcomes you all on this platform. We are very lucky to have this wonderful session today on the topic of microbiome, ocean of opportunities. And before starting with it, I would like to introduce our audience with some eminent personalities who are present with us. First of all, I extend my hearty welcome to Dr. Arvind Deshmukh, President MBSI India. Sir is backbone of this society and always support us to do our best on this platform. Without his support and blessings, these things are not possible. Sir, we are lucky to have you as our mentor, and thanks standing behind us and supporting in all possible ways. Secondly, I extend a hearty welcome to Honorable Dr. Sonjit Singh, State President, MBSI, Chandigarh, Tri-City. The man who works extremely hard and making lot of effort to arrange such panel talks by inviting many honorable scientists from various on a single platform for spreading knowledge. You are the strongest pillar of MBSI Chandigarh Tri-City. Sir has many achievements to his name. He is the former head environmental biotechnology and microbial biochemistry CSIR IMTech. Actually, किसी का mic on. Please mute yourself. Excuse me. Please mute yourself. Hello.
Yes, speak. Bhupinder, speak. Hello, Bhupinder.
Okay. Hello, sir. Am I audible now?
Yes.
Okay, sir. Okay, sir.
Introduce Dr.
He
Soche.
was, sir has many achievements. Okay, sir. Okay. So finally, on behalf of MBSI Chandigarh, I would like to welcome our keynote speaker for today's section, Dr. Yogesh Suche. Dr. Yogesh Suche is currently working as professor at School of Arts and Sciences at Azim Premji University, Bengaluru. He started his career as scientist, National Center of Cell Sciences, NCCS in Pune, India. After working on different aspects of microbial, especially bacterial diversity of various ecological needs, his first area of research is now succession in in international journals like FEMS Ecology and International Journal of Systematic and Evolutionary Microbiology and Microbial Ecology. He is also on editorial board of prestigious journals like Current Science, PLOS One, etc. He is fellow of Maharashtra Academy of Sciences, Indian National Sciences Academy, and National Academy of Sciences, India. In 2009, he was given a responsibility of establishment of microbial culture collection. It is now known as National Center of Microbial Resources. It currently holds more than 150,000 bacteria and fungi in its collection, recognized by WFCC, World Federation of Culture Collection. MCC is also recognized as International Depository Authority and is designated repository by Ministry of Environment, Forest and Climate Change, Government of India under the Biological Diversity Act 2002. In September 2018, Department of Biotechnology extended the mandate of this repository to include antimicrobial resistant microbes. After successful career at NCCS for 32 years, he joined Azim Premji University from 1st March 2022. He is associated with SKAN Trust as advisor. He is also on the member of the executive board of International Society of Systematics of Prokaryotes and World Federation of Culture Collection. We are very lucky to have Dr. Yogesh with us. And now, without much delay, I would like to call Dr. Yogesh to start with his topic. That's all from my side. And thank you for listening me. Over to you, sir. Over to Dr. Yogesh.
Thank you, Bhupender, for that nice introduction. And yeah, so I'll share my screen. I think you can see it now, right?
Yes, sir.
Yes.
Yeah, and yeah, can others mute their mic because there is some disturbance from somewhere?
Please mute your mics. Hello. Please mute your mics. Close your mic.
Yeah, okay. So considering the majority of the audience here is students, I'm going to talk about what opportunities microbiome offers to youngsters and elders alike. So, I mean, at the onset, I'm extremely thankful to Dr. Swaranjit Singh, who is a good old friend of mine, and also Dr. Deshmukh for giving me this opportunity. And since this is being held under the auspices of Microbiological Society Tri-City, that is Chandigarh, Mohali, and Panchkula, I'm very happy because these places have a very special they hold a very special place in my heart, because though I subsequently joined National Center for Cell Science, I started my career as an independent researcher in Institute of Microbial Technology, Chandigarh, where Dr. Swaranjit Singh was my colleague. And then I left it after a year or so and joined Pune. So Chandigarh and IMTech always holds a very special place in my mind, and I'm very happy to give this talk under the auspices of Microbiological Society of Tri-City. And yeah, I can see that there are many youngster, even undergraduate students here. I'll keep my talk simple, and the major thrust of my talk is going to let these youngsters know the ocean of opportunities that they have in future, and they really don't have to depend on looking for conventional jobs when they are doing microbiology. So that is going to be a major thrust of my talk. Now, there was some problem. I was unable to change the slides. I'll do it once again. Yeah, so. Yeah, now I can change it, fine. Yeah, so when we talk about microbes, there are basically three categories. One is bacteria, the other one is eukaryotes, and third one, eukaryotes include protozoa and fungi, and third is viruses. So these are the three microbes. When we are talking about microbes, we talk about these three types of organisms. And when we are talking about microbiome today, we are going to talk about largely about prokaryotes, but to greater or lesser extent, what I'm going to say about bacteria is also going to be true for other group of organisms also. When we look at the human body, it is surprising that there are something like around 25 trillion human cells in our body. The bacterial cells are also present in equal number. Earlier estimate was we have 10 times more bacteria, but now revised estimate says that we have at least equal number of bacteria in our body, and majority of them are present in the gut, as we shall see it later. And these parts I'm going to talk about it more when we during the course of my talk. So majority of these bacteria are present in our gastrointestinal tract. So when we talk about these 38 trillion bacteria that are present in our body, so out of these 38 trillion are present in gut, and small fraction is present in other organs also. And in the GI tract, obviously they are present in the entire GI tract, but the number of cells present in different parts of GI tract is different. So stomach, being acidic in pH, harbors hardly 10⁻³ CFU per ml, as against that colon harbors the highest density of 10⁻10 to 10⁻12 CFU per ml. So density varies across the GI tract, and not only the density, but also there is a local population variation. So different parts of the GI tract, you will find different densities and different type of microbes to be present. And the microbiome has assumed a great importance in last decade or so, because it is thought to be the biggest thing in biology after human genome, with extraordinary human health implications. And it is believed that it can provide simple, sustainable, and safer solutions for human health, which were not possible earlier. It is a very powerful tool for personalized therapies. So human genome and microbiome, when they are used together, they give you a very powerful tool for personalized medicine. And there lies the importance of microbiome. And many important people, including some Nobel laureates, have emphasized the importance of microbiome. Lederberg, then Julian Davis, they all have emphasized the importance of microbiome. I'm not going to read out those sentences. You can just have a look at it. And the importance of microbiome is so much that all important journals in biology covered microbiome as the cover feature in many issues. And today you pick up any journal in science on, at least in biological and medical sciences, on any subject, I can bet you that there will be at least one paper related to microbiome. So even distantly related journals on psychiatry and these things, they will have at least one paper related to microbiome. So that's it. I mean, this is to prove that the microbiome has assumed a great importance in science nowadays. This part I have already said that this was the earlier estimate that we carried 10 times more cells. But the important point to be noted here is that number of genes that are contributed. So human genome contributes only 23,000 genes to our body, but microbes, total microbes present on all the surfaces of body, they contribute several thousand genes to our body. And that's why they are very important functionally. And overall, if you look at the gut harbors the largest density, but other organs also harbor microbes. So there is a skin, vagina, mouth, nose, and many other organs. So upper respiratory tract, placenta, breast milk, they are all known to carry specific groups of microbes in them. And more organs we know to now we know to harbor microbes. I mean, I would just like to mention skin a little more, because although the total number of microbes present in the skin is much less than the gut, but overall the number of species or the diversity of microbiome in skin is as large as that of the gut. This is primarily because the skin microbiome is not uniform all over. As one can expect, the forehead, which is little oily and exposed to outside air, will carry different type of microbes as compared to, for example, armpits or groins, which are moist and not exposed to outside air. So the skin microbiome, there is a tremendous variation locally, and thus there is an, in terms of number of organisms, it is as diverse or probably more diverse than the gut microbiome. So this is something which we should remember. Then I will quickly tell you that why we started talking about microbiome so much in the last decade. So microbiology is a science which is more than 300 years old, but even at the start of microbiology, people knew this phenomena, what is called as plate count anomaly. And what the plate count anomaly means is any sample you take, soil, water, your own tooth scraping, you try to grow them on a culture media. Supposing there are 10,000 cells per ml here, you try to grow them on a culture media, any culture medium you use, you'll at the most grow only 100 cells per ml. So out of 10,000, only 100 cells we grow here. But here you can see 10,000 cells per ml. And that is what is counted as plate count anomaly. And out of 10,000 here, only 100 can grow. People tried many ways to grow this remaining 9,900 using different culture media and all, but with very limited success. But people were always looking for some ways to know what are these 9,900 bacteria that we are unable to grow in the laboratory. What is their nature? And the breakthrough came by the discovery of this particular molecule, 16S rRNA gene, and a pioneer work by this scientist called Carl Woese in 1970s. He proposed and proved that 16S rRNA gene can serve as a molecular clock. In other words, it helps us in accurate identification and classification of bacteria. So once that was happened, once that was accepted and proven that 16S rRNA gene can be used as a molecule for accurate classification and identification of bacteria, then it was possible to use this particular molecule. Even if the organism cannot be grown, this molecule can be used for the identification of bacteria. Of course, that was not possible. That was not possible immediately in 1970s. One more discovery had to happen to make this possible, and that was the discovery of polymerase chain reaction. So polymerase chain reaction enabled us to amplify the 16S rRNA gene from the bacteria. And as you know that in polymerase chain reaction, a single DNA molecule can be amplified many times. So if you do it first round, second round, third round, if you do 30 rounds of amplification from a single molecule, you get few million molecules. So what it means is that in your sample, even if there is a single bacterial cell, you can still get few million copies of 16S rRNA gene of that particular bacterium, even if you are unable to grow it. Even if you have extracted DNA, from the DNA, you can amplify the 16S rRNA gene. And so that made it possible to know which are these organisms that we are unable to grow. And of course, the second step was sequencing. So once you amplify the 16S rRNA molecule, you need to sequence it. And then by doing sequencing, you identify which type of bacteria are present. So earlier procedure was to use 16S rRNA amplified gene. Earlier, people used to do cloning for before going for sequencing. But with the advent of modern day technologies, the cloning was no more required. And not only that, the cloning followed by sequencing had its limitations because it had limited capacity. Very few sequences can be done. It was laborious, time consuming, and expensive also. But the advent of these what are collectively called as next generation sequencing technologies, which are very high throughput, very fast, and cheaper when done on a larger scale. So these technologies enabled us to do a large amount of sequencing at a very cheap rate and in a short time. And if you want to look at the example, at the beginning of this century, when it was proposed to do a human genome sequence, it was to cost 15,000 crore rupees and 11 years. But with the advent of these faster and cheaper sequencing technologies, today we stand in a situation where the same sequencing can be done in less than 24 hours and for just $100. So which means that the the same technology can be used to study bacterial diversity. And if you look at compare the sizes,
11 years back, it is about 15,000 crores. Ipa one it is about $100.
hello. If you compare the sizes, human genome is something like 3 GB in size, and microbial genomes are around 5 to 6 MB in size. So, which means that the in the same cost and time where in which you can do single genome sequencing, single human genome sequencing, you can do 50 to 60,000 microbillions. Can you please put off your mute yourself? There is a lot of disturbance. So the same time and cost in which you do a single human genome, you can do 50 to 60,000 microbial genomes. So that triggered the science of metagenomics, and a lot of microbial ecosystem then could be studied using metagenome because by doing sequencing, because what it needs to be done is extract the DNA and do the sequencing of 16S rRNA gene. You get huge amount of sequence data so that you can study the diversity and you can go deeper and you can study the different type of organisms that are present in the given ecosystem by studying their 16S rRNA gene sequence. So that kind of triggered the thrust in microbiome research. And typically, if you want to look at the way studies are done, there are two ways. One, as I said, that you can amplify the 16S rRNA gene, sequence the 16S rRNA gene, and then using this sequence, you can classify the organisms and find out taxonomic profiling of the bacteria. So what are taxonomically you can identify which are the different organisms present, which give you a lot of information, but still a limited information. Because you are sequencing only one gene from about 3,000 genes that are present in bacteria. As against that, if you do the whole genome sequencing of the bacteria, that means you do much deeper sequencing and try to get the whole genomes from the bacteria that are present in the ecosystem. You can get much better understanding of capabilities of this bacteria, because you get not only taxonomic profiling, but you get their functional genes also. So you get to know which are the different functional genes are present in those organisms, and then you can study how these genes from different bacteria might be interacting with each other, how they act as a community. So this information you get from what is called as shotgun metagenomics. So these are the two major ways in which the ecosystem could be studied, and each has its own merits and demerits. So 16S rRNA sequencing can be is cheaper. You can do taxonomic classification. As against that, the whole genome metagenomics is you can get entire communities, and since you are not limited by a specific gene, you get viruses and fungi also in metagenome analysis, but it is expensive in terms of doing sequencing, and also in terms of computational power required, it is very expensive. As the science advanced, this was metagenomics. It further led to, as you know, the central dogma of molecular biology, DNA to RNA to protein. So in addition to protein, the technology is developed to study RNA, that is transcriptomics, and proteins, that is proteomics, and the end products of the metabolism, that is final metabolome. So today, all these multiple technologies are available to study the microbiome and its function. And people use these technologies as per the need. One or a combination of technologies are available to study the microbiome. So I have already told that humans are superorganisms, and they harbor microbes everywhere, nostrils, external body canal, hair, mouth, GI tract, skin, penis, vagina, everywhere we have microbes. And typically the microbes are acquired from the mother at the time of birth. So when you have a vaginal delivery, the microbes are acquired by the baby, one from at the time of delivery from vagina, and secondly from the breast milk. The baby acquires microbes. If the babies are born by cesarean section, then of course they don't get microbes from the mother, but they get microbes from hospital environment. And then such babies are prone to develop many other diseases, allergies, and diseases like obesity and diabetes in the later part of their life. So having vaginal birth and breast milk is very important for the baby's health. So as the baby's diet changes, there is some change in microbiome after solid food. But then at the age of 3 to 4 years, microbiome becomes constant. It changes then under only under disease conditions. So different stages of life, different activities affect the microbiome. And so as I said, it is stable between infancy to aging. I mean, adulthood, it is stable. As you start aging, the microbiome again changes. And today, a lot of emphasis is being done to study the microbiome of elderly and to promote healthy aging by alteration of microbiome. So, that is one of the major interest in microbiome. And this part, I have already said the densities of bacteria in different part of the body. But and as I said, that the microbiome remains stable throughout the life, and it is altered in the disease. This is the point which we are going to come very soon. So today microbiome is known to have effect on many aspects of human life, development, blood flow, immune system, most importantly, energy utilization and store, production of micronutrients and fatty acids, short chain fatty acids, metabolism of the drug which we take orally. They also known to provide us protection against the orally ingested pathogens. They block colonization with pathogenic bacteria, and we understand a lot of molecular mechanisms of that. So newer and newer role of microbiome in our health is coming to be known. So when we talk about human health, now earlier we used to talk about host genetics and lifestyle. Today we also include microbiome in that. So these three together, our genetics, our lifestyle, and our microbiome, they three of them taken together determine our health. And microbiome alteration in disease has been shown in now many diseases. I'll revisit that again, but major diseases are obesity, inflammatory bowel disease, liver diseases, many other infection diseases, diabetes, atherosclerosis, metabolic syndrome. Many such diseases it has been shown that there is a microbial dysbiosis. And for that reason, I mean, as I said, there are many global microbiome initiatives. So each country had its own microbiome initiative and studied the microbial community structure of individuals in the country, their population. They have studied the healthy microbiome. And this is something which I was telling you some time back that when we talk about microbiome and dysbiosis, there are three kinds of microbes present in any organ, not just gut: symbionts, commensals, and pathobionts. And as long as there is a homeostasis, we are healthy. If this balance is disturbed by some reason and pathobionts increase, then there is a dysbiosis resulting into disease. And this balance can be disturbed by many other many factors. One is genetic factors, other one is microbiome modulators like antibiotics and pathogens, and environmental factors, most important being diet. So if proportion of pathobiont increases, there is a dysbiosis and disease. So if we want to go back to healthy status, obvious thing is to reduce the proportion of pathobionts. And that is what the microbiome based therapies try to do, that they like to push back this balance to the homeostasis by reducing the number of pathobionts. So now reducing the proportion of pathobionts can be done in more than one ways. One way is to kill the pathobionts, other one is to increase the number of commensals and symbionts. So all the microbiome-based therapies are directed towards each of these approaches. And this has been studied in large number of diseases, but most important ones are type 2 diabetes, cardiovascular disease, inflammatory bowel disorder, non-alcoholic fatty liver disease, obesity, autoimmune disorders, metabolic syndrome, cancer, autism, and many neurological disorders are also now shown to have microbiome. There is a lot of studies nowadays on gut-brain axis, where it has been shown that your intelligence, your mood, your memory, all this is affected by your microbiome. So again, one way to control your brain is to alter the microbiome. By altering the microbiome, you can increase your memory, you can change your mood, and you can also to a certain extent control the neurological disorders. So from all these aspects, microbiome is assuming more and more importance. And these are some of the classical studies that have shown the correlation between disease and microbiome. So this, I mean, I'm just giving you one or two examples. There are hundreds of such examples. So it has been shown for obesity that if you put the microbiome from an obese person into the mice are obese. If you put the microbiome of lean person to the mice remain lean. Same is true, and since then, lot of studies have been done on obesity and microbiome, and in general, the observation is that in obese individuals, microbial diversity decreases, microbial gene count decreases, organisms like Oscillospira, Erkeliaceae, bifidobacteria decrease, organisms like Roseburia, Prevotella, they increase. So this is the overall observation for obesity. But these are region specific. So what I want to emphasize here is that specifically Prevotella, though it is found to be increased in obese persons in other studies, in India, the situation is different. In India, the Prevotella is high in healthy persons and it is decreased in obese and diabetic individuals. So there are regional variations. Same is true with diabetes. It was shown in case of gestational diabetes. That is a woman, there is a pregnant woman, there is a condition called gestational diabetes, where the women are healthy in the first trimester and the same woman becomes diabetic in the third trimester. So when you put the microbiota from healthy woman to the mice remain normal. When that same woman becomes diabetic in the third trimester, you put the microbiota in the mice become obese. So this has been very kind of conclusive proof that there is a correlation between diabetes and microbiome. And in the last 10 years, the research has moved much forward. Not just diabetes, but microbiome is shown to have a role in many complications associated with diabetes, like diabetic nephropathy, retinopathy, microvascular complications, reproductive complications. So all this has not only associations have been shown, but it has also proven in the animal models, these effects of microbiome have been shown. So the science of disease microbiome correlation has moved dramatically in last 10 decades. And that's why there are, I have already said that there are many major microbiome initiatives across the globe, and microbiome is seen as a tool for precision medicine. So microbiome manipulation can be used to prevent the disease, for the diagnosis of the disease, for the therapy, and also for the personalized nutrition. So diet microbiome combination can be used for this. And a very classical example of this is a study done in Israel population a couple of years back. And I'll just say it in quickly in very simple words, that here there were some 100 individuals were selected, and the study was done for 15 days, where they were allowed to eat and do whatever they want, but again there is some disturbance. Can you please switch off your mic? So they were asked to record everything on a mobile-based app, all their activities, the food that was consumed, the time when it was consumed, the exercise, sleep, everything was recorded in a mobile app. And using CGR, that is continuous glucose monitoring, using CGM, the blood sugar levels were monitored continuously. And at the end of 15 days, all that data was compiled together and analyzed. So what they found was that your blood sugar is dependent on not only, I mean, the same amount of food atom would result in different levels of blood sugar in different individuals, depending on who is taking it, that is genomics, and also the combination in which that is taken, the time of the day that is taken, and the microbiome. And based on that, they developed again, they developed a mathematical model, and they showed that they can predict the blood sugar level rise in an individual after consuming a particular food. And the model is accurate only when you use the microbiome information is that. So this is an example of personalized medicine, where you can decide the diet of individual based on his genomics and also the microbiome. So the dietary plan for diabetes or for reducing obesity or for anything cannot be universal. It is very individual dependent based on the genomics and microbiome of the person. So that is something which we should remember. And nowadays, more and more people are using microbiome information for this predictive medicine. So I already said that microbiome is a very important contributor to the health. So which are the ways microbiome-based therapies are being done? Of course, the crudest way is use the fecal microbiota transplantation. So you transplant the fecal matter of the healthy individual to the recipient by using fecal microbiota transplantation. But that is rather crude and ethically not acceptable. So people are trying to use microbial consortia or even the lyophilized fecal matter. Further, people have also generated single isolates called kind of probiotics, and then modifying these microbes to give them some unique properties, and also the bioactive compounds derived from these molecules. So there are more than one ways in which microbiome therapies can be used. And the first therapy was fecal microbiota transplantation, which was used in case of Clostridium difficile diarrhea. So this diarrhea has a very high mortality because it is hospital-acquired antibiotic resistant. And when the mortality was more than 90%, but when the fecal microbiota transplantation from healthy individual was used, the success was more than 90%. So today, the fecal microbiota transplantation is used not only for Clostridium difficile diarrhea, but it is also used for diseases like colitis, IBD, Crohn's disease, not only abroad, but it is being used in many smaller towns in India also. So, as I said, that you can use this microbiome in many ways. Fecal micro transplantation is the crudest and the easiest way, but then consortium, then single strain, and then finally the bioactive compounds produced by the gut isolates. And I'll quickly show these slides. If you look at the market today, the base status is you will find that all these are being used. All these approaches are being used for microbiome therapies, and in many cases, even bioactive compounds are also available. So I'll quickly go through this list. It will tell you the for which disease the microbiome therapies are present in which stage of clinical trial, because as you know that if something has to be used for human use, it has to undergo different clinical trials. So as you can see, the list is very long. Many such microbiome based therapies are under different clinical trials right now. So I mean, this is just to give you an idea about the scope of the field, that many such therapies are under different stages of clinical trial, and this list will tell you the pure compounds that are developed from the microbiome that are being currently under different clinical trials. So this is a purified chemical derived from gut microbiome, which is used for the therapeutic purpose. So you will see that the such drugs are available for not only for Clostridium difficile diarrhea and ulcerative colitis, but also for cancers, metastatic melanoma, cancer, obesity. So purified compounds from gut microbiome are also going to be available very soon. Some of these have reached the phase 3 clinical trials. So that means they will be in the market very soon. So that's the reason I want to emphasize that the market for microbiome based therapies is very high, and there is a tremendous scope to do that. So the microbiome therapy for Clostridium difficile was approved by FDA also in the beginning of last year. These are the diversity of compounds that are produced by gut microbes, a large list. So again, I'll not read all of this. And this is an example where the microbiome can produce anti-cancer drugs. So 6-aminopurine is a known cancer drug, anti-cancer drug. But in this case, what was done, or this was done in mice on melanoma. In this case, they used, instead of this purified compound, they used organism that is producing this compound, and they got the success. So these are the kind of applications that microbiome-based therapies have. So the market for microbiome is very huge. Currently, it is $269 million, but its growth rate is 31.1%. And by 2029, it is likely to reach $1,370 million. And it is in the area of therapeutics as well as diagnostics. So that is the market potential that microbiome research has. And microbiome startups is a very emerging sectors. And that's where students like you have an opportunity to come up with innovative ideas and make it a business model. If you look at the 5 top biotech startups which are developing microbiome analysis solution, you will find that 3 of these are for human microbiome, 1 is for animal microbiome, and 1 is for plant microbiome. So microbiome based therapies are not only for human, but they are also important for plant and animal sciences also. So there is an enormous potential for microbiome based business, and these are some of the youngsters who started microbiome as a business. Open Biome by Mark Smith, Vedanta Biosciences by Bernard Ole. So these are just few examples from a large number of examples that are available. And these startups are in variety of areas: oral health, skin disease, intestinal health, drug discovery, dietary supplements, genomics, clinical diagnostics. So the sky is the limit. And here could be some novel applications of microbiome. So again, studies have proven that the professional athletes have a different microbiome than normal individuals. So microbiome can be used to increase the endurance of the person. And this also has been used successfully by some people by transplanting fecal microbiota of a professional athlete to a normal individual to increase the stamina. And one of the reasons for increased stamina of professional athletes is that they have a higher proportion of Veillonella in their gut, which degrades the lactic acid in the muscle and reduces the feeling of tiredness. So this again has been proven. This is an interesting example where a microbiome-based product came up with a simple observation. So everybody must have seen the horse playing in the dirt, but nobody thought why so. The answer to why so, somebody thought about it, and he found that the soil contains ammonia oxidizers, and the ammonia oxidizers from the soil degrade the ammonia in the sweat so that there is no smell. And then that person developed these deodorant sprays that contains this ammonia oxidizing bacteria and degrade the ammonia in your sweat so that you don't smell. Again, the Botox therapy is very common for beauticians. They inject Botox in the skin to suppress the wrinkles. But today, skin probiotic based on microbial consortium is available, which as a replacement for the Botox therapy. I have already told you that your intelligence, your memory, all this is correlated with the microbiome. There are more and more studies coming up that the people who live long have a different microbiome. So microbiome can also be used to increase the age span. The microbiome also can tell you your lifespan. There have been studies which have shown that within a period of 10 years, you can predict the lifespan of an individual by natural death. So the studies are have done on that. Studies are also done on forensic application of microbiomes, where the microbiome analysis has been shown to identify the individuals. So the microbiome of the chair, back of the chair or the seat when we are sitting is same as that of the individual. In a conference room, the microbiome of the room is mixture of all the individuals present there. So these facts have been used to prove the presence or absence of particular individual in a given room. So that way microbiome has many applications. And India, as we know, we are very diverse by biogeography, by diet and ethnicity. So our diversity of microbiome is as much as the entire world. So entire world together, and we are on the other side. So we have been studied Indian microbiome for last few decades in health and disease. I'll quickly show you. So it shows that the microbiome composition of Indian population is very different. So I'll just come to the main point. If we compare that with Western, African, and other Asian countries, our gut microbiome composition is very different, and that is primarily because of higher abundance of Prehotella and Megasphaera in our gut. Same way, if we look at we also studied the alteration of microbiome in diabetes, and we find that under 4 different conditions, that is healthy, pre-diabetic, newly diagnosed diabetic, and those who are with diabetes for more than a year, the microbiome is very different. And there is a transition from healthy to pre-diabetic to diabetic. And then after the drug, again, there is a change. So the point here is that microbiome alteration can be used for 2 things. One is to predict the disease, capture the disease before the symptoms develop, and subsequently to either reduce the severity of the symptoms or the onset of disease using microbiome-based therapies. So there is a more and more scope for such applications. And in this study, what we have also shown was that if you look at the blood chemistry, there are certain parameters which are associated with microbiome. So when we look at the Akkermansia and Prehotella are positively associated with antioxidant capacity and negatively associated with pathophysiology of diabetes. So they are good bacteria. When they are present in higher number, antioxidant levels are high and diabetic markers are low. So, and these facts can be used to control the disease. We have also studied the other diseases like chronic obstructive pulmonary disease, celiac disease, inflammatory bowel disease, and kidney stone. And kidney stone, again, we have found that not only we have found that the gut microbiome of those with kidney stone is different from those of healthy individual. We have also found a kind of probiotic bacterium from healthy individuals that can help dissolving the kidney stone without need for the surgery. So these are some of the important applications of microbiome based therapies. And finally, what is in for? I mean, how we can whether the information is available for the Indian population or not, because if you want to use microbiome based therapy successfully, we need to have information on Indian population. The information available from Japanese, American, Korean population, Chinese population is not going to help us. So that's the reason we had started this study 4 years back, wherein we had looked at 4 different biogeographic regions and 17 communities, 200 individuals from each community, and we initiated to study the microbiome of these 3,400 individuals. We collected a lot of information about their diet, demography, lifestyle, plus their Ayurvedic Prakriti was also determined to find out if there is a correlation between Ayurvedic Prakriti and gut microbiome. So that was a study that was initiated by NCCS. The study is getting concluded now, and NCCS spearheaded the study with the participation of All India Institute of Medical Sciences, SRM Institute of Medical Sciences, IBSD in Imphal, TDU in Bangalore for Ayurvedic Prakriti estimation, and University of Pune. So the results of this study will make the for a certain section of individuals, there is the genome sequencing also has been done. So this study will generate information on genome, lifestyle, and microbiome, which would put together will help us to understand these diseases, the disease and microbiome association better, and then India specific solutions for many diseases can be developed using this information. And this is where I would say that young students like you have tremendous opportunity looking at the examples of what people have done in other countries, especially in USA and Europe, that they have come up with microbiome based solutions for the variety of disorders, including neurological disorders. The same way, the information that is going to be available from this Indian microbiome project can be used by you youngsters to find out India specific solutions for diseases. And I mean, here you can achieve many things in one go. Here you can not only you can help the people by by not curing their diseases but by reducing the severity of the diseases. You are at the same time you are helping the people, doing something good for the public, and at the same time you are earning money. And there are very few examples where you can do these things together, helping the people and making the money for yourself. And then you are not looking for somebody else to give you a job, you are starting your own enterprise. So that's why I wanted to emphasize that there are tremendous opportunities for you in the coming years to come up with microbiome based solutions for our country. It's again an example of Make in India for our people, our microbiome solutions. So I think you all should take good advantage of this. Just about what is being done at SCAN Research Foundation. So here also we are working on healthy aging and neurological disorders. And again, the aim is to find out microbiome solutions for healthy aging and reduce the severity of neurological disorders. And this part, I mean, I'll just want to show that a simple discovery of basic study to look at the microbiome of the mobile phone led not only to discovery of novel organisms, but also led to the discovery of a startup coming up with the idea of developing antimicrobial film for the mobile phones. And this another example is Shrikant, who worked at NCCS with me on a stratospheric microbiome, but then he joined my lab, where again he is working on microbiome, and he was also responsible for developing COVID-Self, that is a self-COVID detection kit. And this is an example of youngster in Bangalore, Piyush Bhanu. So he is again developing microbiome solutions for variety of human diseases, including diabetes. So these are some of the examples of youngsters who have come up with microbiome solutions for human health. And in order to expand this work, SCAN needs a large number of young professionals with experience in microbiome. So specifically, we are looking for those with PhD and with microbiome experience. So either any one of you or your friends have the background and PhD, either PhD in microbiome metagenomics related area or experience in working with anaerobes, please do approach us. This is my entire team, my collaborators, and thank you very much. Yeah, I think we have time for some question answers.
Thank you very much, Dr. Yogesh Soche, for the excellent talk on microbiomes. We learned so many new things from you, from the talk, and it will help the youngsters take up research projects on microbiome. I have just small 2 queries.
Yes.
The microbiome is the first line of defense on our skin. Supposing there is a cut and all that, the microbiome is the first line of defense of the skin, whereas the skin is the second line of defense. What do you say about this?
No, in between your sound was breaking. So I didn't get the questions clearly.
Okay, I will talk again.
Yeah.
Can I repeat now? Can I repeat now?
Yeah, please.
Can you hear me?
Yeah, now I can hear you. Yes, yeah.
Okay. The microbiome is the first line of defense of the skin.
Right.
And the skin is second line defense. So what is your comment about this microbiome of the skin?
See, unfortunately.
Can you hear me?
Yeah, I got you. Unfortunately, the skin microbiome
I'm saying that microbiome
is
is the first line of defense of our skin.
Right, yeah. Yeah, I got it. So, unfortunately, the skin microbiome. And
there's some problem in the connection.
Can you hear me? Hello?
Yes.
Am I audible?
Yes, I can.
Yeah.
Yes.
Yeah.
Hello, I can hear you.
Yeah. So what I was saying was that.
Yes. Yes.
Yeah. So what I was saying was that unfortunately, the skin microbiome has been very little studied because of the complications involved in that. It is a typically low biome sample. So the skin microbiome in relation to the defense mechanism has not been studied in greater details. But what has been shown is that during the infections, during certain skin diseases, the microbiome of that particular location has been changed. So that part has been shown, but the how the skin microbes act as a protection for infections or any other disease, that has not been studied in greater details as much as it has been studied for the gut microbiome.
Right, right. So this is a new area of
Right.
research.
Yes.
People can do research
Definitely, yes.
on
Definitely,
the
yes.
skin
Yeah.
microbiome.
Yeah.
Yeah, yeah, yeah. That's all I want to ask.
Yeah. Yes. Yeah.
Yes, yes, yes. So anybody wants to ask question to Yogesh, you can ask.
Good afternoon.
Yes, sir.
Prakash Salami this side.
Yes.
Any questions by the audience?
Sir, can you hear me?
Yes, okay, Prakash.
Yogesh sir, can you hear me?
Yeah, I can hear you.
Thank you very much.
Good afternoon.
Yeah, just I wanted to know about this scan working on FDN.
Yes, we can hear you. Yes.
What type of research is going on and what are the actually biomarkers you are looking for?
No, see, scan, we have started the research activities.
Survey type of study. Is it right now you are doing some survey type of study or you are looking?
No. Currently, the scan is doing, scan is developing cohorts to longitudinal cohorts to study how to use microbiome as a predictive model for the neurological disorders. So they are establishing 10-year cohorts of large population and then use these cohorts to predict onset of neurological disorders.
Wonderful. Other thing was actually with reference to non-culturable microorganism which we are now estimating through NGS. Does development has happened with reference to their cultivation?
Sorry? You mean to say no enough papers have done for their cultivation?
Yeah.
See, there are a lot of people trying to use variety of methods for their cultivation because it's a rather bold statement to make in front of the students. But a concept has evolved over the last few years that classical method of pure culture of bacteria is an anomaly. In nature, no organisms, no bacterium lives as a single pure culture. They always live as a community. So, and that is a reason probably the microbes don't grow, many microbes don't grow in pure culture. So people are using co-cultures of 2 and more organisms to study the these uncultured microbes, and there have been a lot of success in being able to grow some of these so-called uncultured microbes using this co-culture approach.
Yeah, like next generation probiotics, if we have to cultivate the Akkermansia, we have to so from that angle, as you said, that they may not be able to grow alone. But in a combination or in association with other organisms, it is possible to cultivate them.
Right.
The last one actually is you were talking about this endurance, performance endurance using microbiome. How long actually we supposed to take, or how many days or years?
Sir, those studies, publications, the study that was done, that was, I mean, see, it's not permanent. The effect is temporary. So the person had received the microbiome FMT, and then for whatever shorter period the person had increased endurance. That's all has been shown so far. So FMT is not does not bring out a permanent change in microbiome. It is transient.
The other thing was the complexity of microorganisms. How they are actually dependent on other microflora also to see the effect, beneficial effect.
Sorry, again I missed it. What did you say?
Now the complexity of a microorganism. when we look for the performance of one or other culture, how complexity matters. That is what I was wanting to know.
No, no, again, I mean, I'm unable to get the question. There is some problem in between. I'm missing some words.
Okay, okay. I was actually asking with reference to microbial diversity.
Yeah.
When we look for a performance of a particular potent culture associated with suppose endurance, right? How the involvement of other microorganisms are
Yeah,
also
see, again, this particular case, when we are talking about endurance, it's a consortium that was used. It was a FMT, so it was not an individual culture. So, but when you are moving from consortium to single isolates, I mean, those some of the examples which I showed, all the standard these things to show the functional evaluation of that microbes are done.
yes, sir, yes. Thank you, thank you.
Yeah.
Dr. Namita will ask now any questions if she has.
Yes, sir, I'm interested. Good afternoon, Yogesh, sir, and everyone. Sir, thank you so much for your excellent lecture. Sir, I'm working here in the Department of Microbiology, Mohanlal Sukhadia University, Udaipur. And I'm working on microbial diversity of the goat milk samples and the diversity of lakes. So I have, sir, two questions. First is related to like, hello. Hello. Sir is there?
Yeah, I'm here.
Okay, sir, I am working on the microbial diversity. So my first question is that we have identified, isolated some microbes and we have sent for sequencing. And what we have observed that 16S RNA sequencing is not 100% correct. Like in case of E. coli and Shigella, the 99% sequences are similar. So apart from the whole genome sequencing, what other method we can use to differentiate or identify the microbes?
No. See, you did 16S or you did whole genome?
16S RNA sequencing.
So no,
we
see,
have
for
done.
enterobacteria, 16S definitely does not give the
Correct estimation.
correct identification. So people use some additional genes to correct species level identification for enterobacteria, Pseudomonas, Bacillus. There are some additional genes that are used to get accurate species level identification.
Okay.
So many groups, there is this problem of 16S RNA not being able to give you the complete species level
identification. Okay. Sir, can you suggest some institute where we can go for whole genome sequencing and they are accurate
NCCS, NCMR, they do additional gene identification and also whole genome sequencing analysis.
What will be the approximate cost for whole genome sequencing, sir?
I don't remember right now.
Okay.
Because everything is given on the website. Everything is given on the website. Yeah.
Okay. Yeah. Thank you so much, sir.
Yeah.
Sir, I think you know Dr. Rahul Jain. He is my friend. He he once told me about you that you are in. Dr. Rahul Jain currently he is doing postdoc.
I know.
Oh, he told me about you and your passion about microbiology. So he was telling that you can ask for, because sir, we are in state university and in 2018 we have just started the work. And currently I have one UGC project, and then I have one more question is there, sir. In your one of your slide, it was mentioned that is this stool sample is the correct estimation for gut microbiota. Is it there?
Yeah, because stool is a kind of mix of everything, and in many specific local diseases, the local microbiome changes, and that we have shown it in our celiac disease work. We have shown that actually the biopsy taken for the diagnosis gives you better correlation of the disease than the stool microbiome.
Okay. So in one of project with one of my colleague in zoology department, we are working on the fecal microflora and their antimicrobial resistance of some birds, some water birds. So if we are collecting fecal samples, so can we claim that these bacteria are related to gut microbiota of that
Yeah,
blood?
see, that is not a problem. If you're taking a fecal sample with all precautions, then yes, they represent the gut microbiome, but not
All.
yeah. Some this thing may not be represented in the fecal this thing.
Okay, okay. Thank you so much, sir.
Thank you. Yeah. My video is jumping. There's a reason I'm putting off the video actually.
Sir, can you write your mail ID in the chat
I
box?
had already showed that in the
Displayed.
slide.
I think I have not copied that.
Okay, I'll write it in the chat box also.
Okay, okay, sir. Thank you.
One you have made now just now.
I'm writing it in the chat box also.
Thank you very much for the good questions.
Thank you.
Very good, very good. Thank you very much for the nice answers to the questions put up by Prakash and Madam Namita. Now I request Bhupinder Sharma to say the word of thanks. Bhupinder?
Yeah, so I have put my email ID in the chat box.
Hello,
Thank you, sir.
Bhupinder. I think Bhupinder is not there, Yogesh. So I thank you very much for the excellent talk you have
My
just
pleasure.
given.
My pleasure.
And we have learned a lot from your talk.
Thank you.
And the connection was not very good, but
Yeah.
I've discussed with you over telephone many things.
Sure. Sure.
And
Yes.
Dr. Hamida B also will discuss with you many things about microbiome. And thank you very much, everybody,
My pleasure.
for attending the talk
Yeah.
by Yogesh.
Thank you.
Thank you so much.
Thank you. Bye-bye.
Bye. Bye.